This is a production of Cornell University.
And welcome to this symposium in honor of Peter Davies' contributions to Cornell over the last 46 years. My name is Fred Berenger. Back in the last millennium, I was a student with Peter. I was here from 85 to 90. And I'm delighted to be here today to help MC this event. First thing I'd like to do is introduce the chairman of the section of plant biology, Dr. Craig. You want to have a few words?
No, thank you. Okay, I guess my job is to talk a little about Peter and not to talk too long about Peter. I made a few notes to myself to make sure I covered every aspect of my interactions with Peter. I knew about Peter before I knew Peter. I missed him by a couple of years at Yale, but I knew of his eminence in the area of plant physiology. So I was delighted to meet him here when I got to Cornell. Peter was then the chair of, or soon to be, the chair of the plant biology section. It sounds the same, but it's different. It was a smaller unit. I then knew Peter as a member of the faculty of plant biology. And ultimately, I got to know Peter as, of course, as a colleague and a friend. I got to know that he had a remarkable wife, Linda, who was an intellectual force in her own right. I got to know that Peter was a heck of a dad with two extremely remarkable and accomplished children. Their accomplishments exceed those that anyone could have rationally hoped for in life. I'm still in awe of that, like most of my colleagues, it seems. It's pressure on my kids, I guess. I know Peter as a very interesting teacher. He has courage. He was a good and effective teacher in plant physiology for years, but toward the end of his plant physiology teaching career, he introduced a course where he covered major events in biology and based on the popular press and on things that were happening at the time. I sat in on the course. I hope to reintroduce the course. I was very impressed by the courage Peter exhibited in covering issues that are highly controversial at times. I was quite impressed by the guests and the guest lecturers we had in the course, including one of my favorite old Yale professors, who it turns out is a good friend of Peter's, Dick Goldsby. So that was a nice thing about knowing Peter. I also knew Peter in the role as a wonderful host and a fine colleague whom I like very much. And I think today we'll see that I'm not the only one that shares that very positive view of Peter and his contribution to Cornell. And I'm happy to turn it over to the program.
Thank you very much.
Sure.
Thank you. I think we're going to hear a lot of nice things being said about Peter today. I want to thank everybody for coming today, and it's great that you're all here to honor Peter. I see quite a few familiar faces, but some faces I don't recognize. So I think it's fair to say that all of you have benefited from Peter in some way or another in your career. And he's helped us learn and grow in different ways. But if it was just that, I don't think we'd have this seminar here today, because it wasn't just how Peter helped us with our careers and learning and profession. It was the way he did it. And the simple word for it was it was a lot of fun to work with Peter. Peter brought a lot of energy and positive thoughts to all the work that I did with him. He made it fun to work. He was open to new ideas and supported trying new things. He did everything with a healthy dose of humor, and I think his hospitality to the people who have come through his lab over the years is well known. So, and the last thing is, I know that he kept the best interests in mind of all the people he worked with over the years. So, those are my impressions, and I think you'd share those as well. So it's great that you're all here as a way of showing some gratitude to Peter for what he's done for our careers, and also to be thankful for the way he's been learning and collaborating on science, working in the lab, and fun things to do. So thank you very much, Peter. And I'm going to read a little bio about Peter, so hang on there. Apparently Peter got an early head start in plant biology when he took a class in high school, botany class in high school. And his textbook at that time was by Ernst and Daniels, who were two Cornell professors. So maybe it's no circumstance that Peter ended up at Cornell after all these years. And it was at that point that he learned about gibberellins, which had become a lifelong passion of his. Peter was a member of the faculty at Cornell for 46 and a half years, and a full professor for 33 of those years. He retired this January with the titles of Professor of Plant Physiology and International Professor of Plant Biology. Peter holds degrees from the University of Reading in England, Bachelor of Science Honors in Agricultural Botany in 1962, and a PhD in Agricultural Botany with an emphasis on herbicide physiology in 1966. In between that time, he earned a Master's of Science in Plant Physiology at the University of Davis, and he got that degree in 1964. Prior to coming to Cornell, Peter postdoced with Art Galston at Yale University for 3 years. Peter's published over 100 papers on plant hormones and several books and book chapters. At Cornell, he taught plant physiology his entire career, reaching thousands of students who went on in the sciences and agriculture. He also taught plant growth and development with Dominic K. Law for many years. More recently, Peter started a popular course entitled Issues in Social Biology, covering the societal implications and advances in biology, and even more recently joined the GMO Debate, Science and Society, a course on GMO crops, their regulation, societal implications. And he's been doing that with Ron Herring and Dave and Sarah Evanega, who will be speaking today. Peter has had a long history of service to Cornell. He was the chair of plant biology from 1992 to 96, the director of graduate studies for the field of botany from 80 to 83, director of undergraduate studies in plant sciences major from 2004 to 2009, and chair of College of Agricultural and Life Sciences Faculty Senate from 2001 to 2004. He held the position of Jefferson Science Fellow at the United States Department of State from 2011 to 2014, serving as a science advisor in the area of agricultural biotechnology, and has been involved with Cornell's mission of international agriculture. This symposium today reflects Peter's long-term interest and contributions to the field of plant hormones, his more recent advocacy for biotech crop advocacy, and we are delighted to have Jim Reed and Sarah Davidson Evanega and Ronnie Herring, all close collaborators of Peter's over the years, to help honor Peter's career. So with that, I'd like to introduce our first speaker, who is Professor Jim Reed. Jim is a plant geneticist who has identified genes that regulate plant hormone synthesis and perception. He's shown how gibberellins, racinosteroids, auxin, and ethylene regulate plant development from the molecular to the whole plant level. He's currently working on the role plant hormones play in symbiosis. Jim is an ISI, highly cited researcher, and former Dean of Science, Engineering, and Technology at the University of Tasmania. He's a fellow of the Australian Academy of Technological Sciences and Engineering. Jim spent 2 sabbaticals here. I was fortunate to be a student here during one of those and can attest to how much fun it is to work with Jim. But more importantly, Peter and Jim have collaborated over the years and have made significant contributions to the field of plant hormones and plant development. So Jim, thank you very much for making the trek here from Tasmania all the way to upstate New York. The floor is yours. And the title of Jim's talk is Hormones and Plant Development.
very much, Fred. It's absolutely great to be here, and thanks very much for the invitation and the organisation Sarah and Fred have gone to put on the show over the next couple of days. When I was asked to come, I was asked to talk to a title something like this, which was to talk and give an up-to-date version, overview of where plant hormones are at. And in thinking about it, I thought that field is just huge these days, it's just enormous. There's no way to give a contemporary overview of that whole area. So I thought that I would actually focus a little, instead of trying to cover the whole area, on areas and questions that Peter had asked back in his early career, and try to show you how, over the last 30, 40 years, those questions have been answered. So I apologise if some of the data is a few years old, some of it will be quite up to date. But I think it's important to actually address the questions that Peter posed back in the 70s and see where they've gone today and whether they've been able to be answered going forward. Peter's been engaged with plant hormones for a very long period of time. This is the conference of 1967. So he's been working for nigh on 50 years on plant hormones. That's Peter Davies there, and probably about mid-20s, early 20s. So he's been working in this area for a very long period of time. And I would like to define 3 areas of research which I think he's made a critical contribution to over that very long career. There are areas that I think are close to his heart. He's worked extensively in these areas, and he's really been at the forefront of the field when some of the discoveries were made. The first one is perhaps an odd one for somebody to put up. He's been a champion of hormone physiology. And I'd say that because he influenced me in the early days to be interested in hormone physiology. And I think he's done that through being the author and editor of key texts over a very long period of time to enthuse people to actually get involved with the field. And at times, there's been a real need for that. The second area that I'll talk and probably talk more about in terms of length are the growth-promoting hormones, auxins and gibberellins, the things he worked on, and particularly their control of the elongation, shoot elongation in plants. And he did early work where he identified auxin transport patterns and gibberellin metabolism and some of the gibberellin intermediates in the biosynthesis in a number of key species. Sorry, I probably should have said that the first area is highlighted perhaps by this paper, Hormonal Regulation of Higher Plants, with Art Goulston back in Science in 1969. So this perhaps shows that he was interested in this overview, because this was really a large review in Science at that time. The second one is highlighted by this early review, the botanical review, about the current modes of action of auxin. And the third area I'll talk about, and Peter's made a major contribution with, is the genetic control of development, the use of genes to understand development. And in his particular case, it was about apical senescence, and that was before people started to work on Arabidopsis and have all the model species that we have today. So it may, again, appear, why are we saying this? You assume that's the way people do research. No, not that in the 70s. And the key paper there is this photoperiodic control of apical senescence in the genetic line of bees, which he did with Jerry Marks, who was at Geneva, perhaps a mentor as a geneticist, plant geneticist, and Bill Prosting, one of his PhD students. And I think in each of these areas, Peter's made a major contribution. I'd like to show you how over the next little while. My first involvement with Peter came, as I said, through that first area. And that was this text, which came out in 1970 with Art Goulston, presumably based upon that review in Science. And this came out in my final year as an undergraduate student. And so I was introduced to the area through this text. And we used it for a number of years in our third year course in our teaching. More recently, he's produced these texts on plant hormones. The title's changed a little bit over the years. There have been various editions of them between 1987 and 2010. And this is really the major graduate level text, in my opinion, in the field over the last 20 or 30 years. There are chapters by the experts in the field have been invited to write this text. Peter put it together. You can see his interest in gibberellin because the molecule is on the front cover of both of them, and his involvement in genetics, because here are some drawings, in this case, of various P mutants that affect gibberellin biosynthesis, and in the same way. When Peter started his work, that science paper had these 5 classical group of hormones were known. And since that time, the complexity has expanded, and his texts have expanded along with that, with chapters on new hormone groups like steroids that became defined as a hormone group in 1996, and the latest group, strigolactones, in 2008. There are a bunch of other hormones that have also been identified over those years, polyamines, yes or no, some of the defence hormones, chlorogen's been identified, we know that's the FT protein. Other signaling proteins, like clean peptides, have been identified. These control root shoot information flows during processes like modulation. And there are still hormones to be identified. There's the carotenoid hormone. Carotene is a substance you find in smoke, in bushfires, which affects plant development, but it's fairly clear that there are signaling pathways within the plants, and there's probably a natural hormone that's related to it. So the field has grown, and there is still more to go. Why do we need a champion of plant hormones? Why do we need somebody to stand up and publish the texts, write the books, edit the books? It's hard work, and perhaps doesn't get you the high citations that you may get out of some of the papers in a high-profile journal. And my view is because of some work that happened during the 1980s, largely led by Anthony Trulawas, who questioned whether plant hormones really did exist. He was suggesting that hormone sensitivity was the key controller of plant development. And in the more popularist issues, New Scientist here in 1984, you see that there really was a question. Do plants really have hormones? And what I'd like to show you over the next little while is indeed they do, and show you that the evidence that was put forward against them, and there were 7 reasons given by Trulawas against the role of plant hormones. Some of the key ones are just shown here in black, and they are the failure to correlate hormone levels with development, failure to show localised sites of production, and the effects of the environment on hormone levels were said to be trivial. If in fact hormones really do exist and play a key role in plant development, then we must be able to find evidence that shows that those statements are not correct. And what I would like to show you is that in working with Peter over the last 30 years, we will see evidence that contradicts each of those statements. I won't go back to these statements, so it might be wise just to remember them, but I hope that I can convince you these criticisms that were there in the 1980s have been overcome. And Peter played a key role in championing the role of plant hormones through the text that he edited. To move to the second section of the talk, the growth-promoting hormones, auxin, gibberellins, and elongation. And this is where I've worked with Peter and collaborated with him during some wonderful sabbaticals that we've had, my family have had at Cornell, and that he has had while he worked in our lab back in Tasmania. We used a range of techniques over the years. In the early years, it was great physiology, plant physiology, and a forward genetics approach. As the analytical techniques became better, we used GC, LC-MS techniques, and of course, the last 20 years, the molecular biology. But I hope, again, I can show you that we need all of these range of techniques if we're really going to understand plant development from the molecular level right through to the whole plant level. Why work on shoot elongation? Why look at it as a process that we want to understand? And I hope this graph gives you some idea of why we thought it was an interesting area to work in. If you look at yield in cereal crops, this is just wheat crops, over the period from about 1960 through to about 2000, there's a very dramatic increase in yield. And this increase in yield is obviously attributable to improved agronomic processes and a raft of other things at that level, farming practices. But the key thing was that the breeders produced dwarf varieties, new dwarf varieties that had substantially improved yield. They used the genes, but they didn't know what they did. And it was important to understand how we're modifying our crop plants at the molecular level and understanding, therefore being able to potentially use those same processes in other crops. Why are the dwarf varieties so important? Well, lodging is a major agricultural problem. Lodging is when you get the crop, heavy crop of seeds on the top and heads, and it falls over due to wind, rain, whatever. And the dwarf varieties, especially with good fertiliser application, lodge to a much lesser extent than the tall varieties. And of course, if they're putting less carbon into the stems, they can partition a great proportion of carbon to the seeds. So you've all heard of the Green Revolution, and that's one of the key elements behind it. We wanted to understand what the genes controlling height actually did. So back in the early 80s, I started to collect a whole range of dwarfing mutants, and I was using these as the model. And I could classify them into a range of groups. So there were ones that were short, responded very well to the application of gibberellic acid, and I speculated that they were GA deficient mutants. There were some that were elongated, like SLM, that responded very well to GA inhibitors, speculated they were GA overproducing plants. There were ones that were the same length, elongated, didn't respond to the inhibitor, classified those as GA constitutive. LA and CRY have subsequently been shown to control what are known as the DELA proteins, the production of DELA proteins, which are in the signal transduction pathway for gibberellins. The BR deficient mutants, racinosteroid, they are short, they're dwarf, they don't respond well to GA, they respond very well to the application of racinosteroids. They have quite a discrete phenotype, which they have their leaf colour, leaf shape, stem structure is different from the gibberellin ones. And these are all genes all subsequently been shown to control steps in racinosteroid biosynthesis. There are ones that control the receptor of racinosteroids, and we have other mutants that have a short, bushy phenotype and are involved with auxin metabolism or auxin perception in various ways. So we could guess at what the mutants did, but we needed to prove it. So if I take that first group, the gibberellin biosynthesis mutants, here's the wild type. This is Mendel's dwarf mutant that has been used in the pea crop for at least 500 years. And there are a bunch of other ones that control other steps in gibberellin biosynthesis. We wanted to understand initially what controlled this difference between Mendel's dwarf mutant and this wild type pea. Apologise for this slide. This was the first computer-generated slide I ever had made, so just a copy of it. Obviously, I hope my taste has improved a little since the colour scheme in those days. But in the literature at that time, the explanation for how Mendel's gene was working was that it could affect gibberellin levels, could affect gibberellin sensitivity, it could be affecting auxin levels, another growth promoting hormone, it could be affecting the level of a growth inhibitor, and because of the big difference you get during deethylation of the plants, it could be affecting photoreceptor function, and particularly phytochromic function. So these were the alternatives that were in the literature in about 1982. At that stage, we knew the gibberellin biosynthetic pathway went from geranyl pyrophosphate through oncarine, caranoic acid, GA12, and then out through to GA20. That was established. Peter had worked on that and identified some of the key intermediates in that work. But we didn't know anything more about it. Now I want to talk about this little bit at the end that we added to this pathway and show that in fact these are the steps that are controlled by Mendel's LE gene, and then I'll talk a little bit about this gene. So the first question was, were the gibberellins involved? We took the dwarf mutant, applied GA1 to it, get very rapid elongation. So GA1 strongly promotes elongation, but the precursor molecule, or the molecule that was at the end of the known pathway at that stage, GA20, would not cause this elongation. Bioassays, which is all we had at that stage, suggested that the Mendel's mutant lacked a GA1-like peak that was present in the wild type plant. So that was the hypothesis. And then we started to be able to measure the levels of the hormones using C13 labelled internal standards, and indeed we found that wild type had about 13 nanograms per gram fresh weight of GA1 and about a tenth of that in the mutant. So that suggested to us that instead of the biosynthetic pathway coming through here, GA20, and being deactivated by 2-hydroxylation to GA29, that there was also possible for 3-hydroxylation, this position on the molecule, which was known to activate the relevant molecule. And then it can be subsequently deactivated by 2-beta-hydroxylation. So the hypothesis was that Mendel's gene was blocking, mutant was blocking this step. So we tried to apply radio-labeled GA20 to the plants, and then looked at what was produced, and the mutant GA20 was produced just to GA29, whereas in the wild type, GA20 not only went to GA29, but it also, GA1 and GA8 were also produced. So it suggested we had the right step. The question was, what was the mutant doing? This mutant could be just a regulatory gene, it could be in the structural gene. So we had to try and identify the gene product of that locus, and now we might do this, and we'd probably give a student a month or so to get the job done. Back then, it wasn't quite so simple, it took us many, many years. So, Diane Lester, who was a postdoc in the lab, at the time Peter was with us on sabbatical, Peter was on this paper back, he was with us in 96, 97, and this is work that Diane headed up, but Peter was working with us at that stage. We isolated the 3-oxidase gene by heterologous screening, and then showed that the mutation in that gene co-segregated with Mendel's dwarf phenotype. So here's the parents. Tall has one band, dwarf has a mutation in it, which causes 2 bands, big faint here at 0.2 and 1 kilobase band. And then in the segregating F13 progeny, and in P's that takes about 5 to 6 years to generate, we could show that all the dwarf types offspring had the 2 bands, whereas the tall ones either had 3 bands if they were heterozygous, or just a single band if they were homozygous. So it suggested we'd actually found the right gene. The final proof, of course, is showing that what that gene can do. And here's that gene, the wild type gene, was expressed in E. coli, fed with GA20, and indeed the wild type gene probably can convert GA20 to GA1, but in Mendel's mutant, there was some activity, but only about a tenth of the activity you found in wild type. So Mendel's LE gene is a leaky 3-oxidase gene. And we had other mutants that in fact that were completely null. For example, LE2 that had also. So that's, we've added this step to the pathway, but nobody knew much about the regulation of the deactivation. And I'd like to talk a little bit about the SLN gene. Here it is, here's the wild type seedlings, they elongate very rapidly. And we identified the appropriate gene, and we fed GA1 in this case, it metabolised the GA8 in the wild type, but in the mutant, the GA1 had no activity at all. This is for the genes expressed in E. coli. And the reason I'm talking about that this afternoon is that was the first gene known to affect GA deactivation, known to control the level of the bioactive molecule by regulating its degradation rather than its biosynthesis. So it was an important step at that time. Now, of course, we would take it for granted. We just assume those steps. The question really then became, we took this slender mutant and crossed it to one of these Gibberellin deficient mutants, this one, it's a very severe mutant, NA. What phenotype did we get? Because this one is an overproducer and this one has a block on the biosynthesis. When we produced the F2 seed and looked and selected the double mutant, it was elongated just like the SLN type. And the question was, how could that be? if this has a block in gibberellin biosynthesis, how can this be so elongated? And the hypothesis we came up with was that the NA mutation is site-specific in its action. It does not block GA synthesis in maturing seeds, and therefore these double mutant seeds contain plenty of GA20 when they are mature, and on germination, that GA20 is metabolized to GA1 to cause this elongation. If that's the case, we should get a quite strange phenotype we grow on these plants for longer. If we did that, this is what you get. The double mutant grows on, it's very elongated early on, but then the effect from the GA carried over in the seed wears off, and you get this very short, typical nana or spear dwarf phenotype towards the apex. At that time, we assumed that each of these steps was controlled by one gene. We didn't know anything about gene families controlling steps in hormone biosynthesis. And when we looked at the NA-like genes in peas, we found here's the normal NA gene. It's expressed in the stem, but it's not expressed in the developing seeds. And there's a second NA-like gene that is not expressed in the stem, but it's expressed heavily in the developing seeds. That explains why the seeds could produce GA, but the stem couldn't. And the reason I put it in today is because the NA seeds can produce GAs, and it confirmed the suggestion I made in that first edition of that book that Peter's published on plant hormones, that genes controlling plant hormone biosynthesis can have precise tissue and deposit genetic specificity. We now know that, we take it for granted. Most of the biosynthetic pathways that control plant hormones have gene families controlling individual steps, and they quite often have quite specific sites of action or points at which they operate in the life cycle. It's why a small number of plant hormones can control complex plant development quite precisely. This piece of evidence was important in making that identification. The other hormone, growth hormone, Peter's been interested in right through the early 70s was auxin, and IAA, indole-ferroacetic acid, is a major auxin in most plants. And the question was, if I'm saying GA1 controls elongation, and Peter Piper would have said in the early 70s that auxin was involved in elongation, how do they interact? And what I want to describe is a little bit of work by a colleague of mine who worked in the lab in Hobart for many years, John Ross, and he came up with a hypothesis of how at least one way that they may interact, one of the ways that they interact. His hypothesis was that auxin was being produced in the apical bud and the young leaves, where it was loaded onto the basic petal transport stream for auxin and flows down the stem. And that in the elongating internode, auxin controls the level of GA1 by increasing the conversion of GA20 to GA1 and blocking the deactivation of GA1 either from GA1 to GA8 or the precursor from GA20 to GA29. And he tried to test that hypothesis to see. I'll try to just describe it as this experiment that we need to look at. He had three treatments: an intact plant, plants where you decapitated just below the apex, plants that you decapitated and then reapplied auxin in lanolin paste to the cut stump. And he measured the IA level in those tissues, and if you decapitated the plant, the auxin level dramatically reduced, and if you put auxin back on in lanolin paste, as you would expect, the level of GA was elevated as a triplet. The question was there a subsequent change also in the level of GA1? If you look at this panel, you find almost exactly the same pattern as you find here, supporting John's view that the change in auxin levels seen in these treatments are affecting GA1 levels in the shoot or in the stem. How's that occurring? Well, if you look at the two genes I described earlier, the LE and SLN genes, the GA3 oxidase and the GA2 oxidase genes. The intact plant, in LE, wild type, it's highly expressed, it's decapitated, it's down-regulated, via auxin back, it's up-regulated, and the reverse happens for the deactivation gene. It's low in the intact plant, elevated in the decapitated plants, and then goes back down when you apply auxin back to the cut stump of the enzyme. And as you can see, GA1 endogenous level follows that pattern. So the point from that is that the two growth hormones have been, a lot of work has been done treating them separately. They interact, and one is operating through the other in that particular instance. The auxin is affecting gibberellins. Another question that was posed by Taravis and posed by Peter over a long period of time was the interest in how light controls elongation during denucleation. Which hormones are involved? In the mid-1990s, it was considered to be the racinosteroids were proposed to be the ones that are involved. But it could be the gibberellins, it could be the auxin. I'll try to show you some results and show you that it is in fact the gibberellins that are the primary cause of this early change during denucleation. And if so, how is the plant perceiving the environment? What photoreceptor is involved? I'll try to answer that question as well. If you remember, Taravis suggested that the environment didn't or wasn't affecting hormone levels to any great extent. I'll try to show you that's not the case. I got interested in that work when I was here on study leave back in 1989 with Fred, and in Peter's lab at that time he had some wonderful transducers that could measure the change in plant growth at very fine intervals, and so you could actually measure the change in growth over those first 2 to 4 hours after exposure to red light. So in dark, the plant's wobbling on here, and in red light, continuous red light, its growth rate is reduced to about a quarter of that in the dark. So we want to know how that happened. So I looked at, again, these same couple of genes, the deactivation gene, here in the dark, it's low, and it's elevated within half an hour of transfer of light, and then stays up for a couple of hours, and then drops back to normal after about 4 hours. And the reverse happens for the Mendel's LE gene, the synthesis gene. It's high in the dark, within half, and there's two dark controls here just to make sure the safe lights weren't causing a problem. Within half an hour, it drops down, exposure to light, and then gradually builds back up to normal after about 4 hours. So there's a very rapid transcriptional change over those first half hour. Does that mean there's a change in GA or in the hormone levels? And indeed, here you are, the hormone level after 4 hours in red light. In the wild type plant here, it's dropped to about 1/10 of what it was in the dark. And if we look at a phytochrome B mutant, a Phy B mutant, the difference is the same. But in a Phy A mutant, the difference has been removed. There's no significant difference, suggesting that the environment is affecting elongation by being perceived by phytochrome A, and then that changes the level of the GA1, the hormone, which then results in the change in elongation. So Phy A is the photoreceptor involved, and the regulation of light by light is occurring at the transcriptional level. And just to show you this, if you're interested in phytochromes, here's the cell, the Mansky photograph of the cell, here's the nucleus that's stained for those same cells, and we're looking here, phytochrome, Phy A, been labelled with monoclonal antibody, and in the dark, it's in the round, dispersed through the cytoplasm, it's not in the nucleus. After half an hour of red light, it has formed into speckles and been transported into the nucleus and subsequently shown that that's part of the pathway for action, and then the Phy A is degraded and it's nearly gone by 4.5 hours in the dark. So the pattern of change in what's happening to Phy A is completely consistent with those with other results. Now, I'm talking probably too long, But I was going to take this group of slides out yesterday until Peter told me that he still wasn't absolutely convinced about how autism was being synthesised. So he didn't necessarily believe all the evidence that people had put in the literature. So back in 2009, one of the students in the lab had this diagram. There were at least 5 ways in which auxin was proposed to be biosynthesised, indole-3-acetic acid, from either indole or tryptophan. And the predominant pathway in the literature at that time was through tryptamine and hydroxytryptamine. That was the proposal. And that was based upon the fact that the yucca genes in Arabidopsis had been shown or suggested to control the conversion of tryptamine to N-hydroxytryptamine. And that came from work by Zhao et al. In 2001 in Science, where they published that this is where the yucca genes were operating. And there was a paper in Mays by Leclerc et al. In 2010, also suggesting and providing evidence that this was the major pathway. Well, we tried to have a bit of a look at this and see whether it was the same in peas. And this is one of our grad students, Nathan Tittendale, and he applied tryptamine, and he could not find it getting metabolised to IA. Now, Nathan was a good synthetic chemist as well as being a plant physiologist, so he set out to actually make N-hydroxytryptamine. He synthesised it and looked at its mass spectrum. And when this is the authentic N-hydroxytryptamine, and he checked it by NMR, and when you look at it, here's the major ion, and it fragments to 144. And when you look at, say, the Leclerc et al. Paper, here's the fragment, main ion, and then it fragments to give 106. These are clearly not the same compound. So the suggestion that we were getting was, not only couldn't we find it being metabolised, tryptamine metabolised to this compound in peas, the spectra on which this hypothesis was based appeared to be incorrect. And some excellent work by a couple of major groups, molecular biology groups, in the following year actually moved the yucca genes from here over to this point, controlling the conversion of indole-3-pyruvic acid to IA, with the tryptophan, IA pathway being proposed as the dominant pathway. And now it's thought to be that in Arabidopsis. We want to, again, therefore, check whether this occurred in peas, our model, and John Ross, again, working in the lab, collected genes that control this step, the first step of this pathway, tryptophan to IA, and he obtained mutants in them. And I just want to show you that indeed, and this is in developing seeds, and here in green is the wild type, and here is the mutant in purple. And as you can see, indeed, mutation in that gene in that step has a major effect on auxin biosynthesis. There's a complication because in pea seeds, the major auxin is in fact a 4-fluorinated compound, not just the simple IA, but the same thing is happening for IA at the same time. So it indeed appears that the IPA pathway is the predominant pathway for auxin biosynthesis, not only in some of the models like Arabidopsis, but it certainly appears to be the case in legumes such as pea as well. Well, enough about the growth promoting hormones. The other area I'd like to talk about is the use of genetics to study plant development. And this is just a slide, just grab some information from the literature over the Web of Knowledge and looked at the number of papers that were talking about plant mutants by decade and how they were controlling plant development. And you go back to the 50s and 60s, it was a very, very small number. It was a rare technique, people didn't use it. And it built during the 1970s, has rapidly expanded ever since, until these days, of course, we take it as a matter of fact that it's the technique of choice. But Peter and myself both started working back in the 70s when it was a relatively rare technique. And I'd just like to show you, and this piece of information may only be of interest to Peter, But Peter and during my PhD, I worked on flowering, photoperiod controlled flowering, and Peter worked on genes controlling apical senescence. In the end, it turned out that the genes were exactly the same genes. The genes that control the photoperiod response, whether it be for senescence or flowering in bees, are these genes, HR and SE. Here's a wild type plant, very similar to a wild relative. It branches, it has no real apical senescence, and it flowers very late. If you mutate HR, it becomes if the branching is reduced, the apical senescence occurs at an earlier node, you can't see it here, and it flowers as a quantitative long day plant, whereas this is almost a qualitative long day. And if you mutate SN, the plant becomes early flowering, day neutral, and it will only put on a couple of pods before its senescence. And so these were the genes that Peter and a whole lot of people in his lab had worked on for many, many years. And we all tried to work out what they would do. And it's only really quite recently that we have actually been able to find this evidence out. This is done by Jim Weller, one of my former grad students who works in the lab in Hobart. And he was looking and tried to isolate the HR gene and see what it did. And when he looked at the expression of clock genes, this is circadian clock gene, when he looked at the rhythms, you got the rhythms in the wild type, on transfer to constant light, the rhythm will keep going in constant light. But in the HR mutant, it's dampened out almost immediately. And when you do the same thing and you can transfer not to constant light but constant dark, the rhythm in both types continues. So HR is needed to maintain the rhythm under constant light. When he found that gene, isolated it, he found it was an ortholog of an Arabidopsis circadian clock gene called early flowering 3. And you can prove that. He proved that. Right, here we've got Columbia Arabidopsis, L3, the L3 mutant in Arabidopsis. Here he's transformed those L3 mutants with HR Ps, and as you can see, the wild type HR gene complements the mutant, and they look just like the Columbia wild type. If you transform the L3 mutant with an HR mutant, then they maintain this phenotype, clearly demonstrating that he had the right gene. So one of Peter's genes that was there to control photoperiod response is in fact the L3 gene. It's actually quite an important gene because if you look at phylogenetics of peas, going from wild relatives through the land races in Afghanistan, through to those that are in Mediterranean and Northern Europe originally, you find that there's only a single mutation that has occurred in HR right across that range, and that HR mutant, that single haplotype containing the HR mutation, has become widespread and is the key that has allowed peas to be adapted to spring sowing in high latitudes. So it is actually an important gene, and it's been fundamentally used in agriculture. The other gene I'll go through even more briefly, and that's the day neutral gene SN, so it's wild type mutated with SN. And again, what you find here is the circadian rhythms have been modified by mutation. The mutation's in purple, and the rhythm has been advanced by about 6 hours in that. And when you go through the process and isolate the gene, you can find that SN is the ortholog of the Arabidopsis clock gene Lux. So those two genes that we're controlling, and Peter worked on right through the 70s and 80s about apical senescence, actually control genes fundamental to the circadian rhythm. Is that right, Peter? You convinced?
Oh yeah.
I'll talk to you after.
Good stuff, Jim. Good stuff.
The other thing that now I'm getting a bit more up to date with some of the more recent work I've been doing in the lab. My interests in the last few years have moved to using the hormone genes that we isolated and identified back through the 70s, 80s, and 90s to looking at how they affect and using them to see how other hormonal processes are regulated in plants. For example, the NA gene I talked about, here are the roots of the NA gene. Quite clearly, the roots are smaller and shorter, we expected that, but the nodulation is dramatically, it's almost eliminated, showing that the GAs are absolutely essential for some of the early stages in nodulation. We also look at other mycorrhizal, other symbioses in plant roots. For example, Bastia mycorrhiza. Here, instead of being depressed as it was in that last slide, in the NA mutant, the number of pustules to hyphae is significantly elevated, and that can be reduced or returned to normal by application of GA. So the gibberellins play a key role in regulating both nodulation and the muscular mycorrhizal development. And they're operating in different directions, so we presume they are not operating in the common pathway that controls both those processes. I haven't got time to go into any more detail. Strigolactones, the most recently isolated group of hormones. This is work done by Eloise Boone in my laboratory. So if you take a mutant, the CCD8 mutant is just a mutant in licensed in the pathway for the strigolactones, they branch profusely down the base. Show they contain no strigolactones when you measure them. If you look at fungal structures, substantially reduced muscular mycorrhizal development in the mutants. That's been known for a long time. That's not new to us. But certainly what is new is that they're also affecting nodulation. And we're working on how the strigolactones are affecting the communication between the root and the rhizosphere and how that then results in different levels of nodulation. So that's work we're currently doing. Another mutant we just isolated recently in these, it's been known in other species for some time, is ION2, ethylene insensitive mutant. And as you can see here, here's the wild type, here's the ION2. Nodules all over. It's almost supermodulated, which is a discrete phenotype. So it affects that. That had been determined by other people in things like metacargo before, but what we were interested in is how that effect of ethylene interacted with things like some of our other hormone genes. For example, LK is a racinosteroid deficient mutant because of blocking racinosteroid glycinase. So you get a significant decline in nodulation when you get rid of or reduce racinosteroids, but when you recombine it with the ION2 mutant, produce the double mutant, the implication is that you get an elevation of nodulation back to what you see in the ION2 mutant. In other words, ION2 is epistatic to LK, and when we look at that and try to work out why, it's because this plant, the LK mutant, racinosteroid mutant, has elevated ethylene levels, which would normally be retarding nodulation, and when you introduce an ethylene insensitive mutant, then the difference between the LK and the wild type is effectively lost. So my current work is concentrating really on how the root interacts with the rhizosphere and how the hormones that we've looked at for a whole lot of other processes in the shoot, how they interact to control that symbiosis development within the plant root. So how, and this is important for nutritional gathering by the plant, since the mycorrhizal development leads to increased phosphorus uptake, and the nodulation leads to increased nitrogen uptake. So we're trying to piece together inside the root how the plant hormones regulate these processes so that they are effective for the plant and but don't become parasitic, get to the point where too much carbon is being lost to the microorganism. That's probably at least 5-50 minutes. So this is a photograph taken in our lab back our building in Hobart, where Peter was with us back in 1996, 97. And some of the key people who have done the experiments that I've been going through there, Diane Lester did a lot of the molecular work with the LE mutant, John Ross did a lot of the AUXIN work I've been talking about, Jim Weller is there, did the work on the SNHR, the flowering genes, the clock genes. I think that's probably Ian Murfitt is there, who was my mentor. He was the person who's collected together some of the mutants that I worked on, the SMHR mutants that I worked on way back in my PhD. So he was, if you like, my mentor in getting into the use of genes in colour.
And Ian also did sabbaticals before that.
Well, if you want to go into the story, he did sabbaticals with Jerry Marks up at Geneva and with Cornell. He met Peter at that time, and in fact, Peter ended up being, I believe, my PhD examiner, which I, of course, wasn't allowed to know at that time. They were done confidentially, subsequently, obviously, found out many, many years later. So it's a long and engaged group. And of course, there's been a huge number of collaborators at UTAS, in our lab in UTAS, as well as people all around the world, but particularly in this audience, obviously, Peter at the top and Fred, that I had some absolutely great times out in Kent State doing some work in Dan Cosgrove's lab while I was here over that period of time. So it's been a great, I've enjoyed the career, and the work with Peter has been at the heart of a lot of those studies over those last 20 or 30 years. And I thank him very much for his contributions he's made and what he's given me over that period of time. I should acknowledge the sponsors. Clearly, my university has supported me over a long period of time, and the Australian Research Council has been, it's like your NSF, has been providing funding to allow us to do this work over many, many years. So thank you very much for listening. Thank you.
Thank you very much, Jim, for that whirlwind tour through the history of plant hormones, auxins, gibberellins, bringing us up to speed with some of the latest developments of senescence and circadian rhythms. Those are all topics that have been dear to Peter for many years. So that was a really great talk, very fitting presentation. We're going to take a break in a few minutes, but if anybody has any questions, we'd be happy to entertain them now.
Well, I've got a comment.
As always.
Or comments, questions or comments.
Well, I never believed the tryptamine story anyway. It sounds like MC Stewart saying that. We're coming back to the pathway that was proposed in the 1940s. So you've got...
All I would say is go and grab a text from 2008, 2009, 2010 and see what they were talking about. Yeah, you're absolutely right. There have been evidence and people supporting each of those pathways, and they each kind of had their turn in the sun. I think the difference is we've now isolated the genes in some of those pathways. We've been able to put those put mutations in those genes and place them in the pathway, and actually not only follow a change in the phenotypic development of the organism, but actually monitor the hormone levels and show that change. And that, while we did that in the 80s for GAs and in the 90s for steroids, really has only been in the last 8 or 10 years that we've been able to do that in the same way for autism. So even though it was the first hormone identified, and we really should have known how it was biosynthesized years ago, it's really only in the last few years that it's become, I think, nailed down.
I'm just going to do a quick introduction. First person to really show the pathway of auxin in roots, which is now central, and he also did auxin biosynthesis in roots, my first undergraduate student, Eric Mitchell. You stand up, Eric, please. Who's now come back. Eric is an MD, pathologist, but we nearly got him to go into plant sciences, but medicine called. Right, Eric?
Well, I actually did. There was another student that I collaborated, and we decided that if I went into medicine and got the money, we could start our own lab. But then I got married, and I can't tell you
I'm probably not qualified to ask questions about plant biology anymore. When I left a while ago, Rhabdomyosis has been a key model organism. So I'm interested to hear how much work you've done with peas to elucidate these mechanisms. Was some of that work done with Rhabdomyosis as well?
Okay.
Because genetics are so much.
No, look, there was a period of at least a decade where the field was dominated by the molecular advantages of Arabidopsis. There's no doubt. For example, when we pulled out that GA3 oxidase gene I talked about, Mendel's gene, there had been, we would not have been able to pull that gene out unless somebody had identified previously in Arabidopsis that there was in fact a gene that controlled 3 oxidation. And so we were able to pull it out. So a lot with Jim Wells' work, the recent work there on the flowering genes, as I said, they'd been identified as clock genes in Arabidopsis. We now have a sequence for metacargo. He got the sequence, the equivalent sequences out of metacargo, and then pulled the sequences out of P. We had the mutants. We didn't know what the gene structure was, but he was able to prove it by actually going back and finding mutations in the homologs out of P. So Arabidopsis, in terms of being able to allow us to advance this molecular front, was fundamental for at least 10 to 15 years. We're getting to the stage now where we've got enough other genome sequences that we can start to work from those species in their own right. If you might ask, why do I work on symbiosis? What doesn't Arabidopsis do? It doesn't form symbioses. So the obvious reason, if you want to stay working on your favourite model, is to actually look at developmental processes that are important in agriculture, important developmental processes, but things that do not happen in Arabidopsis. I guess you'd have to say that's why we're now working in that area of my lab. It's an area that you have to have the mutants, and you have to be working in a species that has the symbiosis. And legumes have the advantage. They have both modulation and muscular mycorrhizal. Therefore, they are a really good model for doing some of the basic molecular work. So that's why we're where we are in one sense.
Well, some of us are very attached to peas. I've worked with Peter, so it's good to see that they're robust and being used in the research work. Any other questions? Lots of comments? Okay, we have about 25 minutes or so to have some refreshment and meet each other, so let's take a break and we'll get back. It's my pleasure now to introduce Sarah Davidson Ibanega. Sarah came to Cornell after completing the BA in biology from Reed College, and then went on to receive her PhD here at Cornell in 2009. For her dissertation, she conducted interdisciplinary study combining plant molecular biology with science communication, and focused on the controversy over genetically engineered papaya in developing countries, focusing primarily on Thailand. She says, lured by the great weather here, today that certainly applies, I'm not sure that's anything. Plenty of water, and an unbeatable intellectual environment, who can argue with that? She remained in Ithaca at Cornell University after completing her PhD, and she helped lead a global project at that time to protect the world's wheat from wheat stem rust. Sarah now serves as a director for the Cornell Alliance for Science, which is a global communications effort that promotes evidence-based decision-making in agriculture. She teaches courses on agriculture biotechnology at the graduate and undergraduate level. In addition, Sarah serves as the Senior Associate Director of International Programs in the College of Agriculture and Life Sciences, and holds an adjunct appointment in the section of plant breeding and genetics in the Integrated School of Plant Sciences. Sarah was also instrumental in launching the CALS initiative, AWARE, Advancing Women in Agriculture through Research and Education. Throughout her time here at Cornell, Sarah's interacted with Peter. The first time was as a grad student in Peter's Plant Growth Regulators graduate level module in the fall of 2001. She's gone on to work with Peter in international programs as well, has contributed to lectures for Peter's social issues in biology class, and she's also been on the faculty team along with Peter and Ron Herring that has explored the GMO data. Sarah, with your common interest and collaboration with Peter on his activities related to ag biotech, policy, and public understanding, it's great to have you here today. And your talk is, as you can see, global aspects of plant biotechnology.
Thank you, Fred. It's an honor to be here and to be asked to celebrate Peter's retirement in this capacity. I think this is an area that Peter and I share a real interest in. Peter has been a great mentor and source of support for me, as Fred mentioned, for many years and in many different capacities. Both as a graduate student, he was always that faculty member who made sure all the graduate students had a place to go for Thanksgiving, invited us over to his home, etc. That means a lot to graduate students. And it's been an honor to teach with Peter in more recent years. And of course, the remote doesn't work, right? Let's see.
She might have mine. Batteries.
Okay, and of course, he was also one of our real supporters as a fellow graduate student in Bob Turchin's lab, Ashley McCaskill, and I delayed and procrastinated writing our PhD theses and launched roller derby here. Peter was one of our founding fans, always a great pillar of support. It is a little bit overwhelming and maybe humbling to speak on this particular issue because Peter is probably one of the most, one of the best people in the world to address this very topic, global aspects of plant biology. So I'm a little humbled, but I will launch in here. So I think many of you can appreciate that genetically engineered crops are being grown around the world. 12% of the cropland globally is sown with genetically engineered crops. There's about 10 crops that have been commercialized and are out there. 8 of them are grown in the U.S. The U.S. is the largest producer of genetically engineered crops. We produce the most. Brazil follows, then Argentina, India, which Ron will probably talk a bit about today, and then Canada. You can see in this graph that many of you may be familiar with, there's about 180 million hectares sown to genetically engineered crops today in about 28 different countries. And about 5 years ago, the number of hectares grown in developing countries exceeded developed countries. And of those 18 million farmers growing genetically engineered crops, the majority are farmers in developing countries. However, despite this widespread relative success of genetically engineered crops, there really aren't very many genetically engineered crops out there in terms of diversity. So soybean comprises 50% of that hectarage, maize is 30%, add in cotton and you're at 95%, and if you add canola, that comprises 99% of the total genetically engineered crops that are planted today. And so what this suggests to me is that there's a real lost opportunity to use these tools of plant science and plant breeding to improve some of the important crops that are out there in terms of food security crops and really highly nutritious crops. In a similar vein, too few traits have actually been deployed. So over half of the genetically engineered crops that are cultivated have carried the trait for herbicide tolerance, Roundup readiness, as we sort of commonly hear it, and the other dominant trait is herbicide tolerance. And if you combine those two traits together, you have covered about close to 100% of the crops that are planted out there. So again, too little diversity of crops that have been able to benefit from these tools, and too few traits that are actually in the hands of farmers. And I think, yeah.
Go back. What is the reason for the dip in herbicide tolerance in the last?
The developed resistance is probably overall the number of hectares of genetically engineered crops dipped this year for the first time from 2014 to 2015, and there are of course the herbicide tolerant crops have developed resistance. So we've heard a lot about Palmer amaranth taking over cotton fields, for example, in the South and other pieces of the East. So it has been some movement away from some of these crops in some places.
Actually, Sarah, can I jump in with
Now that we've had the wine, you can jump in all you want. and if we had more wine, we can start to debate whether or not plant hormones exist.
That diagram on this graph is herbicide tolerance only, insect resistance only, and more now are combining the two, and that is stack traits. That means both together. So you'll see stack traits takes quite a jump up as the other two go down. That's because they're growing the combination much more.
Oh, I see. Moreover, there were price changes over time, so a lot of corn land went to wheat. So there's, every year, farmers said, and we don't have any genetically modified wheat.
So I think most people in our society today know the reasons why we don't have more genetically engineered crops out there in the hands of farmers. There's been a lot of controversy around this technology, and unlike the field of plant hormones, not much has changed over the last decade or two in terms of conversations around ag biotechnology. And this is despite that nearly every highly reputable scientific organization in the world has endorsed the safety of genetically engineered crops over and over again for several years, not the least of which is the recent study by the National Academy of Sciences that came out a couple of weeks ago that again emphasized that there's no substantiated evidence of a difference in risks to human health between conventionally bred crops and genetically engineered crops. And they did not find in this study any conclusive cause and effect evidence of environmental problems from genetically engineered crops. Even the Girl Scouts have taken a strong stand for science on this issue when they were pressured a year and a half or so ago to remove genetically engineered ingredients from their cookies that they're so famous for. They took a stand and cited worldwide scientific support for the safety of currently commercialized ingredients derived from genetically modified agricultural crops. And they went on to say that in the future, GMOs may actually offer a way to help feed our ever-increasing population. Go girls. There have been a few studies published that have suggested that there are risks to genetically engineered crops. And I think the most famous of which is the Seralini study. But that study was retracted after it was published in 2012. It got some play. It had major impact on some policy in various countries around the world, and then was later retracted. But it wasn't retracted before, as I say. It had some pretty serious implications, not the least of which was the effect it had on policymaking in Kenya, where there was a ban put into place on the import of genetically engineered crops and genetically engineered food. And despite the fact that the Seralini paper has been retracted and there's a long list of ways in which it was flawed, this ban remains in place. It's also had impact in countries like Uganda, where groups like ActionAid that have been campaigning against the use of genetically engineered crops have cited this study, and I will, and they even go further. So I'm going to play this.
ActionAid is an international charity raising tens of millions of pounds a year to alleviate poverty and end hunger. In Uganda, as a bill was being debated to allow GM crops, ActionAid Uganda ran this radio advert for a week in 2013.
Did you know that genetically modified organisms are organisms made in the laboratory by crossing genes from different species? Such products pose health risks such as cancer, infertility, etc.
A single study linking GM maize with cancer in rats was discredited by the European Food Safety Authority. A different study suggesting a link to infertility was withdrawn.
So this gentleman in the red sweater is the former director of Greenpeace UK. And of course, Greenpeace is an organization that's campaigned against genetically engineered crops for a number of years.
This message is brought to you by ActionAid Uganda, protecting our health, environment, and farmers' right to seed.
Well, that is really shocking to be scaremongering about health risks, particularly cancer, with no scientific justification, me, that is totally wrong.
So I share that example of a way in which the Seralini paper has had impact on policy and decision making in Uganda. But I also share it because I think it's really encouraging that people like the former director of Greenpeace are actually starting to come around and nuance their position on GM crops. And I think we're at a tipping point, and we may be experiencing a turning of the tides, if you will, around the conversation around GM crops globally. But I'm also encouraged by people like this woman in this photograph, Matiya Chowdhury from Bangladesh. She's the Minister of Agriculture there, and she has stood very firm in her support of genetically engineered crops and ensuring that farmers in Bangladesh, one of the world's most vulnerable countries, have access to this technology. So she pushed for the approval of BT brinjal in 2013, and that material was made available to farmers, a small group of farmers of about 20 farmers. And you can see that ceremony here. And this gentleman, Mansour Sarkar, was one of the first farmers, we go back first 20, who received this seed for genetically engineered BT brinjal. So the eggplant, as we call it here. So in 2014, he planted a small quarter-hectare plot of this BT brinjal and was rewarded shortly thereafter with a crop that he could then market as pesticide-free. BT, of course, is the trait that reduces insecticide use in these contexts. So when growing conventional eggplant, he had to spray his crop twice a week. Sometimes as much as twice a day under heavy infestations. And now he doesn't have to do that. And so he sees this both as an economic benefit as well as an environmental benefit. He and his wife and his daughters eat eggplant 2 to 3 times per week. And the money that they earn from eggplant helps him to support his family and importantly sends his girls to school. His neighbor's eggplant at this time, I'm sure he has better seed now, but at this time, when the seed was originally released, he was still growing conventional eggplant, and this crop was riddled with pest damage and not fit for sale or for eating. So the stakes here are pretty high. Eggplant is South Asia's first genetically engineered food crop. It was developed by public sector scientists and really intended for resource-poor farmers like Mansour Sarkar. And his story, I think, is really remarkable because it's a case where one single ag innovation and strong political will on the part of Matiya Chowdhury have really offered him the chance to greatly improve his livelihood and the lives of his daughters, and that could ultimately break their poverty cycle. His story, however, should not be so exceptional, and the case of BT brinjal that we all like to talk about and celebrate should not be so exceptional. There are a lot of products in the pipeline in late stages of development that have been developed not by the Monsantos of the world, but by public sector institutions in the U.S. and around the world that are quite promising. So products like biofortified bananas being developed in Uganda, products like these nutrient-rich tomatoes being developed by a public sector institution in the UK, plant-derived fish oils being produced in Camelina, another project happening in the public sector in the UK, late blight-resistant potatoes, cold cows. So these are cows that have been genetically changed to not grow horns, which eliminates the inhumane process of dehorning, so this addresses a real animal welfare issue. Insect-resistant cowpea. This is a major food security crop in countries of West Africa. And biofortified or virus-resistant cassava, to name just a few examples. So it's my hope that while we in this country sit around with the luxury of debating whether or not we want to label our foods as containing GM or non-GM, this technology doesn't bypass farmers like Jemima, this cassava farmer in Uganda, who in 2016 still struggles to cultivate a crop that is technically under domestication and could use improvement with genetic tools. It's my hope that in 4 years or 5 years, I can be telling Jemima's success story in which she's had the option to choose whether or not she wants to grow genetically engineered crops, and whether she thinks this technology is something that will benefit her family. And that choice isn't made by organizations like ActionAid that are based in well-fed countries like the UK. I think it's also really important, and particularly in the context of Cornell, that we ensure that scientists around the world have access to the tools that they need to innovate to meet the great challenges that we face today. We've seen what's happened with GM, and there's new tools coming along, and we need to make sure that we don't lose access to those tools as well. So we need to make sure that public sector scientists can use these tools to innovate. Scientists like Alison Van Enneman from UC Davis, who's been responsible for the old calf work that I mentioned. Scientists like Chuck Maynard and Bill Powell up the road at SUNY ESF. And they've taken a gene from wheat and moved it into chestnut to protect chestnut from the devastating blight that has, for the last 100 years or so, decimated nearly every American chestnut tree on the planet. So they're using this technique to hopefully reintroduce a near extinct species into the forests of the eastern US. Scientists like Tony Shelton, one of our own entomologists here at Cornell, who's using genetically engineered insects to reduce pest populations for cabbage growers in New York State without the need for excessive insecticides. Again, a real environmental benefit. Scientists like Andrew Kagundu from the National Agricultural Research Program in Uganda, and he's taken a gene from sweet pepper to protect his country's starchy banana staple, the Matoke banana, from bacterial wilt disease. So it's scientists and farmers like these who I've talked about in the first few minutes of my talk that have really inspired us to build the Cornell Alliance for Science, the initiative that Fred mentioned that we're leading here. And our mission really is to promote access to scientific innovation, access to choose to use these innovations as a means of enhancing food security, improving environmental sustainability, and really improving lives globally. And we think biotechnology is an important part of that. So in our first year, we're a relatively new organization, just 2 years old, founded in 2014. And in our first year, we really focused our efforts around this central pillar of access to innovation. But following the success of COP21, we are now launching a new global dialogue around climate change. And central to this dialogue is helping explore how this technology or new precision breeding technologies that are coming online might help us mitigate and adapt to climate change. So we face a steep climate challenge, that of improving the environment, reducing poverty, and increasing food security, while ensuring that the global temperature does not increase by more than 1.5 to 2 degrees. And that's impossible if we don't make major strides in agricultural innovation. So we're out there in the world, our communications team, talking to farmers, to scientists, to consumers, to policymakers about how they think this technology is playing a role in helping them deal with climate change. And there's some really interesting stories out there, stories of adaptation, like the BT brinjal that I talked about. Water efficient maize for Africa is another great case. But there's also stories of mitigation, so projects that are looking at developing nitrogen use efficient crops or methane reducing rice. The other piece of this Climate for Change initiative that we're leading is to continue to use our training programs to develop champions for change. And we have 2 different training approaches with the Alliance for Science. One is a 12-week program that happens here at Cornell. Many of you probably met our inaugural cohort when they came last year. These are champions from all kinds of different walks of life, different countries, 10 countries who were represented last year, who all feel passionate about this issue of ensuring choice and access to improved modern ag technologies. And they want to ensure that they can bring that to their own countries. So who are these people? A few examples. This is Reverend Father Noli El Parse. He was one of our fellows that came here to Cornell last fall. He's a priest of the Roman Catholic Church. And he came to this issue of biotechnology when he asked himself the question, how can I expect people to come to church on Sunday and praise and worship God when they're hungry and mired in poverty? This is Cletmanjwi Masiga from Uganda. He's a sorghum breeder, and he was also one of our fellows last year, and he asserts that every other field, every professional field, is taking advantage of scientific advancements, and there should be no exception in plant breeding. So why is it that the tools plant breeders have, or plant scientists have, are being so politicized to the point where they aren't able to use them to serve his farmers? Or Joni Kamiya, she's the daughter of a farmer from Hawaii, one of the first farmers to adopt the genetically engineered virus-resistant papaya that was developed here at Cornell. And growing up, she saw firsthand how this technology could positively impact small family farms like hers. And she wants to ensure that other small family farms around the world can also benefit from this technology. So that's a sense of who our fellows are. I was going to show a short film about them. I'm not sure if I have time. Yeah? And your job in this film is not only to enjoy it, but to try to find the Peter Davies cameo appearance.
You signed it.
I didn't sign it.
It was a cocktail party.
Right.
How about money?
My name is Joni Kamiya. I'm Father Noli Alparse.
My name is James Foley, Betwalvi Masega.
I'm Marlon Assis from the Philippines.
My name is Vivian Kiwe.
I'm Jason Merkley.
My name is Abud Modesta.
My name is Nassim Mubanya.
When I was a student in college, I became an activist fighting a dictatorship. I was with like farmers and laborers, and they were so poor. So I kept on thinking, how could I help these people?
It's really very painful when you see families that cannot afford to feed themselves at least 3 times a day.
It's ironic that farmers, the very people that till the soil to produce food, are the same people who experience hunger.
We're not just talking about American consumers here that have plenty to eat. We're talking about people in food insecure regions who have nothing to eat but a small handful of rice every day.
My story is Jemima's story. She's one of those smallholder cassava farmers. Cassava, a major food security crop, is under threat by 2 major viruses. This is very devastating.
All the trees being chopped down, all the papayas had virus in it. It's heartbreaking for him. He said, I was so scared, Joan. I was so scared that we lose everything. Traditional ways of farming in my country are not enough to be able to feed the many. And I think the science and technology that is happening in our midst could be a solution to that.
Indeed, biotechnology can be of help to solving some of these problems. And therefore, if this technology is adopted by the farmers, I think it can help them greatly.
And it's my firm conviction that it's a fundamental right for farmers to have access to various farming options.
It seems very cruel to deny that technology to those people who are in such desperate need.
Access to biotechnology is really a social justice issue.
Being a part of the first batch of cohorts of the Cornell Alliance for Science is truly a very rewarding experience for me.
This is a completely unique program. As they say here in America, awesome.
There was really a science to communication, and I knew that I needed to learn that. So when I found out about this program and that opportunity to learn more about how to communicate science, I couldn't do anything but jump at the opportunity.
And there was a completely new approach. They have what they call grassroots approach.
I got interested in this program because it was an avenue for me to improve on my communication skills in the area of agricultural biotechnology.
My participation in this is hopefully to empower me to speak up and get others to speak up in support of this?
Interacting with experts, agronomists, scientists, farmers, policymakers, journalists.
Commitment and dedication can actually help someone to get further and prove a point.
And there's such a rich exchange of knowledge and wisdom, and the friendship. We're becoming like brothers and sisters.
Knowledge of science, because I strongly believe that science can be a key in solving hunger, malnutrition, and poverty. I cannot do it alone. I cannot succeed on my own, all by myself. That's why I realize the need to unite, the need to engage, and the need to empower a network of partners. Right now, I really feel empowered. Right now, I am more confident in engaging the public in scientific undertakings.
Isn't fascination and discovery wonderful?
This is one of the best experiences that I will ever have in my entire life.
So that was the 2015 cohort. They went home in November, and we just selected and invited the 2016 cohort who will come here in August. They're coming from, there's 10 coming from countries across Latin America, 3 fellows from China, and about 16 fellows coming from different countries in sub-Saharan Africa. So I hope that those of you who are on campus will come out and meet them when they arrive. So since returning home, fellows like Modesta Bugu, who you saw in the video, have gone home and they've recruited 5 to 6 additional champions who share her mission of ensuring that Nigerian farmers have access to biotechnology, access to choose this. She's recruited them into her initiative. These 6 champions then attended our short course in Tanzania that we held in March. And then they went home and recruited 2, 3, 4 more people. And so you can see how very quickly someone like Modesta becomes a very catalytic champion for change through this program. So as the Alliance for Science here at Cornell, we're really trying to engage individuals and organizations who share our mission and want to extend and lend their voice to our global collective. And so I encourage any of you who share this mission and passion that Peter and I do to join our alliance. We have now engaged people through our efforts in over half of the world's countries, and I think we're really building something that's important in ensuring that science and evidence guide decision-making globally. That said, the reality still is that too few products have come out of the public sector, and we're still dealing with that situation that only 4 crops have been approved using these tools. And of course, we like to talk about Dennis and the papaya that was developed here at Cornell. That's the only case where something has come out of the U.S. land grant system. And I hope that will change because this is really something that's consistent with our land grant mission. And I think as scientists, it's really important to step up and take a stand for science and not get run over by people who are making a living spreading fear about biotechnology. So we need to invest in the research. We need to invest in capacity building, both in terms of scientists as well as communicators. We need to engage with our communities, and hopefully we can ultimately improve the policy environments that will inspire a change in the conversation around biotechnology, and ultimately more of this technology can get in the hands of farmers. So my appeal to you scientists in the room is to really, look up from the bench and realize that we need to do things differently. So we sort of lost it on GM, and there's all of these really exciting transformational technologies coming down the pipeline that are here, and we need to make sure that those don't get demonized in the same way that GM has. And I think this is where we can all get inspiration from Peter, because he's done a remarkable job in the last few years of doing just this, both on campus, through his courses. It's been said a few times today how much impact he's had in wrestling with and the tough issues with students here on campus through the social issues in biology class and also the GMO debate, and also through his Jefferson Fellowship and through his volunteer work after the Jefferson Fellowship. He's really circled the globe from Latvia to Botswana to India, helping people understand this technology so that they can make their own informed science-based choices about whether it's good for their country or not. So I take inspiration from Peter. I encourage you to join the Alliance, and then I wouldn't be doing my job if I didn't promote our new MOOC, Massive Open Online Course, that Ron and I have been developing in connection with Patrice from Academic Technologies, who's here, and Becca Harrison, who's here in the third row. Also members of the MOOC team. So this is a massive open online course that we've been developing over several months that'll launch September 13th. And anybody, anywhere in the world can take this Cornell course if they have an internet connection and access to a computer. So I'm going to play our trailer, and this is going to be the perfect segue to Ron Harrington's. Oops, maybe I'm not going to do it.
Early in the 21st century, citizens of planet Earth were distressed and anxious. The problem? What to think of the GMO, the science, the politics, the clashing of values. Scary stories and grand promises compete. Contention swells. What are the benefits? What are the risks? How is a responsible global citizen to think about all this? The GMO MOOC team has come together from different backgrounds, bringing different skills. A scientist. David, a behavioral economist. Ron, a political scientist. Sharon, an information specialist. And Becca, a social scientist. They may not always see eye to eye, but together they pledge to guide learners on the ultimate voyage of discovery. What is the GMO, and why does it matter? Embark on this voyage of discovery, and share your views with others from around the planet. Learn how science works, the limits of science, risk, human behavior, and how you can defeat the confusion in your own life. Sign up now and let the Cornell GMO MOOC team help you activate your own information superpowers.
That's been the premiere, right? But enrollment is open, so with that, I will turn it over to Ron. Thank you.
Thank you very much, sir. That was awesome.
As we say in America.
So, yeah, just a quick introduction for Ron, and we'll move on. Ron Herring is Professor of Government, an international Professor of Agriculture and Rural Development here at Cornell University. His field research in South Asia has centered farmer interests, class politics, and government policy. His first book was Land to the Tiller, The Political Economy of Agrarian Reform in South Asia. That was published by Yale University Press. It was on responses, peasant movements for social justice and development. Curiosity about GMOs was ignited by the intense controversy around BT cotton in India. Farmer welfare, sustainability, the crop plagued by a crisis, and the lowest yields in the world. This research stirred him to edit a special issue of the Journal of Development Studies, which is called Transgenics in the Poor, Biotechnology Development Studies, published at the Fulbright Outreach. This interest continued in his work was continued as editor of the Oxford Handbook of Free Politics and Society in 2015, and with Robert Hallberg, the annual review of Resource Economics section of Political Economy and Biotechnology. Ron is now working on his book manuscript entitled Suicide Seeds and Silver Bullets. With Peter's interest in public policy and public perception of driving GMOs, it's very fitting that you're speaking today, and thank you very much for contributing to the symposium.
Can we do a handoff for the little ticker? Well, it's a real honor and privilege to be here. I wasn't told there would be molecules, but otherwise I'm okay. Peter and Sarah, as well as Shantu Shantaram, who's sitting right here, have helped me as a social scientist understand what is one of the more bizarre political phenomena in the world. And that's the GMO debate. When you think about it, it's really very interesting that there should be a debate about biotechnology, and everybody asked, are you pro or anti? And students have decided to drop the course, in part because all the profs are pro-GMO. It's a very bizarre thing. You think about it, there's a course on like, I don't know, computer technology or cell phone technology or something. And people say, are you for it or against it? It's a very bizarre position, but Sarah and Peter and people like Shantu off the Cornell campus have helped me as a social scientist understand this. Now, my position on this is kind of the obverse of Sarah's. Right? Sarah is a very hopeful person, and I cheer on her hopefulness and indeed contribute periodically to being part of a hopeful campaign for farmer choice, informed choice, and defeating misinformation that is contrary to public good. All for all of those goals, and I periodically write such things in that regard. On the other hand, I don't think the picture is quite as rosy, and I'm going to give the flip side. Right? What Sarah told you about the advantages and the possibilities of science communication runs counter to a lot of what we know about science communication and risk. So I'm going to talk more about the flip side. There's a benefit side to biotechnology. There's also a discourse of risk, and that discourse is politically powerful. So I'm doing plant politics. And I hadn't thought about plant politics much till I was asked to be part of this. So here's Michael Pollan, who really thinks plants have politics. Michael Pollan wants to tell us that all these plants have convinced human beings to give them special privileges. Politics is who gets what and how. So all of these plants have qualities like sweetness, beauty, intoxication that have caused human beings to protect them and invest in them and improve them. And of course he periodically backs off from that because he knows a little bit of science. And so he backs off and says, well, actually plants don't have much consciousness, but this is the burden of a botany of desire. And in Michael Pollan's great question of the battle between the trees and the grasses for dominance of the earth, it is human beings that, of course, have intervened on the side of the grasses to defeat the trees. And that is kind of the nature of the development of nature on the planet. Now, plant politics, though, putting Michael Pollan aside, human beings do plant politics. And there have always been kind of demon plants. There are dozens of such things. I didn't know you injected THC, but this is the weed with roots in hell, and there have always been kind of classification of plants, politics of categorization of plants as unacceptable or acceptable. And of course, the British Empire, I did this because of Peter, of course, British Empire profited enormously by using opium as and its progeny for control of much of Asia. But here I want to point out, we're talking about the effects of plants. So both marijuana and opium have definable effects that you may approve of or disapprove of, but we have a new set of plant politics. This is the new set, the GMOs. It's an apocalyptic version. Oh, by the way, Sarah talked about the Alliance for Science. I'm actually wearing the Alliance for Science. And I'm wearing a jacket because Peter, every time I've seen him lecture, wears a jacket. I thought I'd always be underdressed. But I can't wear a tie with this. So anyway, so this is the new plant controversy. And what's important is that unlike opium or marijuana, the entire category of GMOs, what is a GMO anyway? The entire category turns on how the plant got the traits that it had. It's not the products of the plant or the safety of the plant per se, it's the process that produced it. And two of the most important elements here, of course, are the hubris, they're playing God, and they're shoving it down your throat, which is the dimension of power. So that's the kind of opposition. This is a biblical response to it in third edition, by the way. Now, there's the darker side, which is an attack on science. This is biotech genocide, Monsanto collaborators. There's a list of Monsanto collaborators who are targeted for death. And the Nazi legacy of science justification for murder. And this is a health ranger in natural news. Right? So that dichotomy of the natural and the unnatural, and the health ranger who watches out for dangerous things like glyphosate, is part of the discourse now. this is of course the other kind of critique, that GMOs in the third world cause farmers to commit suicide. This is a very, very powerful discourse, and it won't go away. It turns out that it's simply changing its form, changing its shape, but this discourse of farmer disaster will not go away. Okay? So here's our university course, and we took this question on, and it's evolved over time, but you see the different departments that have sponsored this course. And we try to take on the GMO debate to talk about science and society and how science is accepted in society. What are the costs and benefits of technological change? So that's the core of the course. Right? Now, the profs diverge in this course, but the students fail to notice. This is not surprising. Peter Davies says, people just don't understand the science. This is his shtick with the State Department, right? You guys have got to understand the science, and he has huge amounts of science behind him. It says there's really nothing to worry about. No problem here. Right? Ron Herring says it's not about the science, it's about politics. And that the whole debate has more a nation. Remember the notion of truthiness. Remember Colbert, Stephen Colbert? Where they're truthiness rather than facticity. So it's not about facts, it's not about evidence, it's about truthiness. Now, this is a much stronger claim to make right now. Do you realize what's happening to the Republican Party in this country? Right? Has anyone ever stopped to ask whether or not Mexican immigration in the United States is increasing or decreasing? It's decreasing dramatically, right? Has anyone ever asked this guy? Well, anyway, I won't go on about it. But the fact is that the facticity has completely disappeared from the debate. The majority of Republican primary voters believe Barack Obama is not an American citizen. 70% believe Muslims should be banned from this country. So there's an incredible amount of truthiness substituting for facticity. So the first puzzle in politics is, why did rDNA crops become framed as a separate category? Law, trade, social movements, consumer behavior, international relations, where other applications of the same technology were normalized. Pharmaceuticals, vaccines, industrial products, evaluated by product, not by process. Right? So what I want to claim here is that the GMO was a brilliant political invention. It loaded one concept with all of the risk and disutilities that you could imagine. And Sarah mentioned some of them. There's sort of cancer, and there's sterility, and all the incredible numbers of things that GMOs cause, autism, so on the spectrum down. But excludes all of the clearly definable benefits, such as higher farm yields, or in recombinant DNA work in pharmaceuticals, improved products. So, science and politics. What do we know about politics? Science across the board is inherently local. The thing like global warming, classic. Science commits itself to tentative conclusions. It won't give you a bumper sticker. The certainty of kind of bumper sticker epistemology is restricted to the non-scientific community. Scientists are expensive, and scientists are not disinterested, right? Scientists disagree. The inevitability of fringe science. We saw Seralini's work. It's been tremendous. If you ask how many citations are there to Seralini's article about rats with tumors compared to Peter Davies' work on plant hormones, I'll tell you a secret. Seralini wins, hands down, by huge quantities. So scientists disagree. The inevitability of fringe science makes it possible for people to say, well, not all scientists think global warming is happening, not all scientists think it's human-induced. And then finally, policy makes kind of impossible knowledge claims to disprove negatives. No scientist will tell you there cannot be a black swan. No scientist is going to tell you no one will ever discover an allergen in some transgene that was produced, that was put into a crop in the fields. That cannot happen, simply because the nature of the epistemology is science. So you're disadvantaged politically. You know, the environment minister of India, in turning down V.T. Brinjal, said, I want absolute certainty that nothing could go wrong with this plant. And of course, you could never have absolute certainty in any scientific process. then finally, institutions are not trusted. Nina Federoff, probably many of you know, Nina Federoff said in her presidential address to AAAS that I am scared to death of the anti-science movements all around the world. And it's true. Okay, so this is the kind of worry about science is sort of using people as guinea pigs, right? That what we know is based on a knowledge that is not complete. Every scientist, being honest, will admit it's not complete. It's tentative. Tomorrow, a new study could show that we were wrong about something. So this cute kid he's not a scientific experience. Stop GMOs. Now here, this gets back to the Seralini study. People are also convinced that there are thumbs on the scales, right? That professional journals are edited by people who are part of networks, which is of course true. We're all part of networks. I've edited 2 journals. We're part of networks in which we know who to send papers to get the response we want, right? So peer review doesn't necessarily tell us very much. Seralini's work on rat tumors, which is total rubbish, was peer reviewed. In a peer reviewed journal, not a high status journal, but a peer reviewed journal. So this is from the Herald in Edinburgh a couple of years ago. Study suggesting genetically modified food killed rats has been suppressed by lobbying of multinational biotech corporations, environmental campaigners claim. So there's a thumb on the scale that science is produced by the very corporations that benefit from the bad science that they produce. That's a widely shared view of how science works. So let's just think about this division I made between green biotech, which is food, and red biotech, which is medicine, pharmaceuticals, and vaccines, things of that sort. So look at the risk-utility balance. Virtually all of technology change and technology politics revolves around some balance between risk and utility. Think about flying an airplane or surgery, or any of those things. People know the risk, try to figure the risk. You know the utilities, you try to figure the disutilities, and then you make a decision. So the risk in red biotech is balanced by high utility. The doctor prescribes a drug, you may die if you don't take it. The comparative safety of these drugs is vetted by authoritative knowledge, certified experts that work through certified organizations, state science. Right? So we have state science, you either trust the FDA or you don't, but there's a state scientific process that vets certain kinds of safety. There are voluntary acceptance of risk and benefits, unlike biotechnology, in which what's growing in fields, whatever effects it has, you don't sign on voluntarily to be part of that. You don't sign on voluntarily to consume the food that's produced if it's not labeled. So there's a kind of involuntary subjection to risk, which we all know by scientific studies shows that people will enhance their assessment of risk if they believe that they haven't signed on for the risk. They haven't voluntarily subjected themselves to the risk, unlike drugs or surgery or air traffic, right? And so green politics, biotech, GMOs, it's produced a very different politics, but it's quite variable. Now, Sarah presented this map after telling me we should never use this map again because everybody's bored with it. But this is the map, so the world sort of divided between GMO and non-GMO countries. Sarah gave you a nice overview of how few crops there are, how few traits there are. But one thing she didn't say, well, maybe she did say this, but one thing that people often miss is how little of this is direct food crops. Right? Mostly these crops are either for fiber or feed. So you have this remarkable thing where in the EU, all of the farm animals that are used for consumption, all of those animals are fed GMOs, but somehow going through the gut of a pig removes the stigmata of it being a GMO, right? Even the euro is made from BT cotton fiber. so in the EU, there's an odd kind of transmogrification in the world of growing food crops that are transgenic or genetically modified in some way, and the world of safety and non-risk. And somehow animals stand between human beings and the crops that way. But very little of this graph is food crops for direct consumption. Brinjal in Bangladesh being a dramatic example. an exception to that rule, since brinjals are eaten in Bengal 2 or 3 times a day, often. So it's a major vegetable crop. India is the second largest producer of brinjal in the world. So these are major vegetable crops, and but mostly biotech crops do not go into that kind of circuit. So this is sort of when Sarah gave the example from the U.S. Academies of Science. This is a European study, and I think in a way it's good because nobody trusts the Americans globally. Nobody trusts the Americans because Monsanto does the studies, Monsanto has its finger on the scale, da. So this is a conclusion from the EU studies, from a huge set of studies they funded themselves, meta-analysis. There's just no difference in risk profiles between transgenic plants or genetically engineered plants and any other kind of plants. But what's important about that is that tells us, going back to our earlier thing on plant politics, the entire GMO debate is a political debate. The entire classification is political. There is no difference here except that created by political entrepreneurs. But there's no evidence of any difference except that created by political entrepreneurs. So it's not about the science, it's about risk politics. Okay? Now, think about the benefit side of it. We said there's kind of an equation in technology policy. There's a risk, there's a benefit, there's a regulator that weighs the risk and benefits and decides whether we can go forward or not. Think about nuclear power. Very complicated risk-benefit calculation. Think about global warming, think about Fukushima. Very complicated calculations in the risk-benefit. So, in the benefit side of this equation, the benefits to farmers have been quite obvious. Every country where biotech crops have been made available, they've been adopted pretty rapidly. In fact, the adoption curve in India for things like BT cotton is just sort of like a 45-degree angle. Astonishingly rapid adoption, down like 98% of BT cotton in India. So farmers, they have a risk-benefit calculus too. But all adoption of cultivars involves risk for farmers. If you don't adopt a new cultivar, you're taking a risk as well. So farmers are continually at risk. But the GM crops important to American farmers are much less important to European farmers. So you get this difference in the US and Europe because European farmers don't really need, certainly not BT cotton. They don't grow a lot of soy. The corn rootworm doesn't show up very much far north of Munich. So Spain and Portugal are heavy users of BT corn, but not much of Northern Europe. So they have a different kind of utility calculus, and they're protected by government policy that provides them with pretty substantial livelihood independent of the market. And banning GMOs, of course, reduces competition from American products. So there's a different kind of benefit matrix for European farmers compared to Americans. And the final point I want to make here about farmer adoption is that farmers on every continent have taken very high risk to grow illegal crops that are biotech whenever the prices are too high, sometimes the case, or access is restricted. So virtually all over the world you'll find instances of farmers taking up the technology. There's an underground diffusion network for biotech seeds, even when they're not approved by governments. So the benefit side of this equation is pretty simple for farmers, producers. And of course firms. Most firms have made money on these products. Not all firms. They sometimes go out of business, but firms and farmers have some benefits. The risk, how do you calculate risk? This is the hard part. Benefits are easy to measure, and they're very clear to a farmer. The risk side of this equation is completely politically determined, and that's because of what we call Knightian uncertainty. When you can't measure risk, or you don't find any evidence of a specific hazard, there's no specific hazard, and you can't measure the risk precisely, then you're in the world of uncertainty. And that uncertainty means that you have to construct some kind of risk calculus even though you have no hazard. And as I mentioned with the EU studies, there is no hazard demonstrated. So any kind of risk you have with regard to GMOs is entirely anticipatory and hypothetical. Okay? Now, the most parsonage explanation of variance in outcomes, this is classical political economy, right? You weight the beneficiaries. How many farmers are they? Do they have organizations? Do they have access to the ministries? Do they have some political clout? Then you look at the opponents. Do they have large numbers? Do they have resources? Have they mobilized a lot of people? Do they have media access? And then they interact, and some kind of vector sum comes out of a regulator, which we'll just call for now a government. This is critical, because part of the variance around the world is explained by the character of this regulator, that is the political structure of the evaluation of science. So in the EU, you have multiple veto points and multiple veto players. Public opinion about GMOs is about the same in the US as it is in Europe. In both countries, people are very suspicious. The percentage of people saying it's unnatural, it may be dangerous, is roughly similar. Everybody asks the questions in different ways, so it's hard to compare public opinion surveys. But there's not a huge amount of difference across the EU and the United States. The difference is that very small numbers in Europe can make a big difference. You have multi-party governments in which the Greens often have a Ministry of Environment, or have at least a clout with the Ministry of Environment. And those coalition governments can be blocked by the action of very small numbers of players. So there's this regulator who is in charge of agreeing between beneficiaries and the opponents what kind of risk society should take. Now, empirically, around the world, ministries of agriculture have supported biotechnology. Sarah gave you the example of Minister of Agriculture in Bangladesh supported BT brinjal. The Indian BT brinjal, from which the same transgene, it's actually the same genetic event. The Cryogene seed came out of India, donated to Bangladesh. That very same gene was not approved in India, and it was still put on moratorium in 2010. It's still on moratorium in 2016 now. And why is that? Because in India, the Minister of Agriculture did not have say. The Prime Minister did not have say. The entire decision was made by the Minister of Environment. Ministries of Environment all over the world are much more risk averse for reasonable reasons, but also because their political constituency is environmental groups. The political constituency of agriculture ministers is farmers, and farmers expect progress. So they have a different kind of system between those places where the Ministry of Agriculture dominates and those places where the Ministry of Environment dominates. So India doesn't have BT brinjal, and Bangladesh does. Now, since Sarah brought this up, I do have to mention the hazards. You know, we say risk is hazard times incidence, or hazard times exposure. The hazards in India are very well documented. They're very well documented. The spraying of brinjals presents hazards to both the consumers and to the people who are doing the spraying. We have evidence on it. It's very clear that the current system of producing brinjals is quite hazardous. I talked to some farmers in Maharashtra 2 years ago, and I was asking about BT brinjal, and they said, yeah, we spray 2 or 3 times a week. And I looked kind of horrified. They said, but not up to 5 days before harvest, right? Because all of these bottles of poison say do not spray within 5 days of harvest. And they said, oh yes, up to the hour of harvest if we have to, if the worms are there. And I looked horrified again. And they saw my reaction. They said, oh, but we wouldn't feed this to our families. This just goes to the market. So, but there's a genuine hazard involved in the current use of current ways of growing brinjal that would be alleviated to a large extent by growing BT brinjal. But that's, the regulator was, of course, risk averse rather than opportunity. So I've kind of already said this, and the reason that science has a hard time is Rumsfeld's unknown unknowns, the black swan phenomenon, right? And in human behavior, we have tons of evidence for this. Loss dominates gain, right? So if you're healthy, you walk in the supermarket, you do not want to do anything that might cause some small, no matter how small the risk, you don't want to take a risk if you don't have to. And so you get very powerful risk politics. I guess the most impressive way is, and science has no answer to this, right? So there is no answer to uncertainty. If you don't know, you don't know. I want to give one example of this in the vaccine debate. You know, most people that can read would know that all of the official science says there's no risk of autism from a vaccine. But suppose there's a shadow of a doubt. Suppose there's a tiny, tiny chance that the studies aren't adequate, or they haven't done enough studies, or they'll change the studies next month. You have absolutely no risk in not vaccinating your children so long as herd immunity is maintained in your area, right? So if herd immunity is maintained, even though you're free riding on everybody else's vaccinations, there's no risk to you. But the fact that herd immunity is being lost in many areas and vaccination rates are declining, this indicates that this risk discourse is very, very powerful, even though it's a tiny, 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 tiny risk. One other example is interesting. One of our former students did a study of raw milk consumption. I said there are 13 more states allowing raw milk consumption now than there were like 10 years ago. And I was kind of stunned by this. I thought we were getting more rather than less health conscious, right? CDC has these big block capitals, do not drink raw milk. 90% of the illnesses from drinking milk come from raw milk. So I asked this student who comes from a dairy farm in California, I just asked her about her farm and her feeling about raw milk. And this study said, this is terrible, this is really horrible, all these people drinking raw milk. I said, have you ever drunk any raw milk? She said, well, of course we all drink raw milk on my farm, but it's my cow, right? So she trusts the raw milk on her farm, just like the farmers growing brinjal that I talked to. They're not going to use poison on the brinjals that they eat, they're going to use poison on the brinjals for the market. Okay, so I mentioned cell phones early on. Right? It's a classic case. There's no debate about safety of cell phones. Actually, from the time cell phones came out, there has been a certain amount of debate. This is from Science, May 27th, that's last week or 2 weeks ago. Tumors in rats from cell phone radiation. That's non-ionizing radiation, so you think, how can there be tumors? Well, no one's quite sure. But the critical question here is, and as usual, it comes with the caveat that studies of rodents can mean little for humans. So a rational person says, I don't care how many rats you've killed with a transgenic crop and how many studies, it may not apply very well to humans, and in fact the rat is not a very good model for human physiology. So we had this problem of cell phones all adopted. Everybody adopts cell phones because its utility is so high that even as the risk goes up, people's risk-utility balance stays on the side of utility. You don't see any campaigns against cell phones. There are no demands to label cell phones, although in Scandinavia they do label the output radiation. But basically, we've accepted the risk-utility matrix for cell phones, and we don't for GMO food. Okay, so what divides politics? There's no single dimension. This is unusual. Somebody studies international politics. They're not variations on a north-south pattern, developed, underdeveloped, rich, poor, or democratic, authoritarian. They cross all of those lines. There is no dimension to this global rift, unlike all the rest of international politics. So I want to talk a little bit about risk politics in food. So we've said there are a number of claims about GMOs and their risk, they claim their benefits. Talk a little bit about food, because we know from prior to GMOs that pervasive anxiety about food drives politics. This is a long-standing issue, and it comes up every year. The CDC at one point said 5,000 people a year die of food safety infraction in the United States. They've reduced that to 3,000. These are epidemiological estimates. Nobody knows how good they are. We know that lots of people die from problems with food safety in the United States. There's a distrust of institutions, and of course in the EU, this was especially true after mad cow disease and several other catastrophes where state science got it wrong. Okay? And then there's this capacious political opening for risk entrepreneurs. If you have high anxiety, and it's almost impossible to convince people of safety, then you have a capacious political opening for risk entrepreneurs. Here's one. So what do? We rely on epistemic brokers. This is one of my favorites. This is FoodBabe. People know FoodBabe? Yeah, okay, so FoodBabe, it's time to break free from the toxins in your food. Now, FoodBabe, she looks very friendly for somebody who's taking toxins out of your food. But this is what I call an epistemic broker. People all over the world are in networks looking for cues about who do you trust. nobody out there reads Cell or Nature or Science, right? So what they're looking for is epistemic brokers that they can get cues from that crystallize the information they need. And so here's epistemic broker one, the food broker. Here's a science guy. Who are you going to trust, right? So, here's a science guy, this kind of dingy, ill-lit room talking to small crowds with some kind of cheesy PowerPoint, you know, and he's got a rat here that looks like it's a Seralini rat, you know, and he's got this engagement from the grocery store scary thing, right? So that, they're going to put this guy, and look at this, right? He represents the Department of State, goes around the world trying to convince people. And it just looks kind of like a gray bureaucratic group. Are they really on the side of food safety? I don't think so, right? So, so this is the broker number 2, and there are many like things. And here, look, this is Love the Food Babe way. So who are you going to trust in this kind of competition? We're in social media networks. Who are you going to follow? You're going to follow Peter, the food babe. Well, food babe does very well, okay? So does she. This is Vandana Shiva. Now, her advantage is not being as attractive as the food babe. Her advantage is that she claims to be a scientist, which is not true. She has the advantage of indigeneity, the authority that comes from indigeneity. Indian female woman right there amid some kind of grain crop. So this is a different kind of epistemic broker, but you'd be astonished how powerful she is. She gets $50,000 a lecture in the United States. Astonishing. What am I being paid today? What? Oh, yeah, right. Peter, how much do you get paid? I'll have another chip. This is Vandana Shiva, one of Peter's favorites. She's the one that tells us. Then finally, we said institutions. There is empirically evidence that the perception of mass publics compared to the perception of scientists is diverging a bit more than it had before. So if you ask scientists about global warming and GMOs, the divergence has increased and the public is less scientific than it used to be relative to the scientific community. So the FDA, the power of Monsanto dominates the FDA, so in fact we can't trust anything the FDA says, right? And of course there's these incredibly powerful campaigns. This sweet kid, and he's being killed along with the bees, and he's in a bee protest, and the GMOs are killing all the bees. It's such a sweet picture, and you don't see his face, and maybe sort of, okay. So let me sum up the risk politics. There are very few general laws, because there's great volatility of time, place, and population. If somebody had told me 2 years ago, or even 6 months ago, that Donald Trump would be heading a major American political party, I mean, seriously, how can that happen? So politics is inherently unpredictable, okay? But celebrity predictably drives a lot of politics. Think the food babe has this treatment. She has absolutely no background in science, biology, food, anything, but she has this kind of presence. She's a celebrity, yes. Why is Paris Hilton important? Well, she's a celebrity. Why is she a celebrity? Because she's a celebrity, right? And that's the Donald Trump phenomenon. Okay. So this volatility of time and place means we have no general laws. It's hard to come up with a governing law that makes any sense for politics. But we do know that loss dominates gain. And secondly, there's pervasive anxiety about food, which just creates political openings. History matters. Compare nuclear power. If you're doing a risk-benefit analysis pre- and post-Fukushima, very different kind of analysis. Or think about the Malthusian anxiety. No countries in the world oppose the Green Revolution. Right? Green Revolution plans to resolve world hunger were sold on the grounds that we had a Malthusian crisis, people were going to die of starvation, and we got public science involved, a tremendous increase in productivity of our major food crops. In fact, 8,000 high-yielding varieties were introduced, if you count all of them all over the world. Huge numbers of improvements in plants, and that Malthusian anxiety, the benefit of doing that overcame, there was no risk discourse. Why was there no risk discourse? There were no transnational organizations, right? The transnational advocacy organizations did not exist at this time. There was no Greenpeace, there was no Friends of the Earth. None of those people were in the political game. they are now dominating the political game in many places. And finally, I don't know what happened to my brain here, but I forgot the last point, which is that structure matters. So who these groups can approach, where they have a chance to have influence, where the veto points are, where the choke points are, varies from country to country. The transnational advocacy networks have been tremendously effective at blocking biotechnology all over the world. For example, so this is a Greenpeace. We're not lab rats, and every single sign is the same. They're all middle-class people. They're all in cities, and they had a tremendous impact on what 9 years of field test, lab test, scientific consensus about the Bt brinjal will be good for farmers and good for health and the environment. It was blocked, right? So we have to think about this, the power. Now, Greenpeace has been essentially delegitimized in India. It's been essentially thrown out of the country in an important sense, and Cornell was blamed for this. I in particular blamed for getting Greenpeace out of the country. Okay, so how might things change? I don't want to end on a pessimistic note. One way, here's our problem for how things might change. Risk narratives are as difficult to stamp out as garlic mustard. Everybody here pull garlic mustard? I mean, it doesn't go away. Every time you break off a stem, it forms a new plant. I mean, it's just hope. It's like Asian zinc foil. So okay, so it's not going to happen. These risk narratives will not go away. The Terminator gene hoax is still floating around India, and it was used in a high court decision in the Philippines. It's not there, even after we knew that farmers were underground taking this technology and pocket breeding new varieties of Bt cotton. Even after that, and even Vandana Shiva, she was complaining about the failure of the government to stop all these farmers from breeding new hybrids of Bt cotton, dozens of new hybrids of Bt cotton, while talking about the Terminator gene, right? So how do you breed new varieties from a plant that kills itself? Anyway, so how am I thinking? You're not going to wipe out these narratives. These narratives are very, very durable. So one quick story. There's a book by Heath and Heath called Made to Stick, about how ideas are stuck in people's minds. And they have an example of the last chapter. They say, how do you unstick a stuck idea? And they say, actually, we have very few examples. Now, Sarah's working on the other side of this. Sarah may be unsticking things, but on the other side of this, he said, the only example they could come up with was Ray Kroc, who ran McDonald's. I think that's the right name. He ran McDonald's, and he was accused of having worms in his hamburgers. He said, how can he have such cheap hamburgers? Well, he puts worms in his hamburgers. Ray Kroc goes on TV, and he says, now here we have hamburger, it's 39 cents a pound. Here we have worms, night crawlers. I bought them from the local bake shop, $2.99 a pound. I can't afford to grind up the night crawlers and put them in my hamburgers. And they said, that's an example of coming unstuck. To me, as businessmen are out there to make money, Ray Kroc would not be grinding up night crawlers to put into his hamburgers. So this must be, so he kind of, McDonald's went from there to success. Anyway, so this is just a kind of a counter. I firmly believe in Sarah's narrative, but how might things change? So, and this is my other example, the autism business. This is unbelievable. By the way, this was also promoted by someone like the Food Bank, Jenny McCarthy, who was a Playmate of the Year or something like that. But this was also supported by celebrities. Morgellons disease, the House of Representatives voted to give $1 million to CDC to investigate a disease that's an invention by a psychotic 17th century Spanish physician. And there are websites for Morgellons disease. CDC said this is a web hoax, we don't want to deal with it. And the Congress said, we've got to spend $1 million investigating Morgellons disease. So there's a report. Morgellons disease says we can't find any evidence for Morgellons disease. We're sorry, we just wasted $1 million. So the power of these kinds of narratives should not be underestimated. And of course, if you think about it, suppose you have this kind of narrative, taking apart every piece of that. You know, Monsanto doesn't really own our government. Is it really poisoning our food? And it's really just taking that apart requires a kind of activity which is so much more demanding than this bumper sticker. Okay. So maybe increased utility for consumers. So fill in your favorite botany desire here. Let's say sugar without calories or something like that. Or game-changing, non-plant applications of biotech. And here, I think the combination of climate change, which Sarah mentioned, and also non-plant applications of biotech may turn this corner. And the Zika virus is kind of my favorite. And the Aedes aegypti carries 4 really, really deadly diseases: yellow fever, chikungunya, dengue, and now the Zika. If you can wipe that little mosquito out, you know, no great loss to the human race. It's invasive anyway. You might have a game changer for increasing a massive global benefit at the risk of getting rid of a pest. Thank you.
Thank you very much, Ron. And just hurriedly switch from Sarah to Ron. I wanted to open up the floor for any questions or comments. Thanks for those great talks. These are, first, I think many of you share this. It's a very vexing problem. It's frustrating to all of us. It's great to see people so impassioned, trying to do something about it. So kudos to you guys. Good luck with the future. It's really important. So, any comments, questions? Randy.
Yeah, I'm neither pro nor con GMOs, but I noticed that Sarah's slides were labeled with Alliance for Science, and Ron's T-shirt was labeled. I assume because you're proud of the Alliance for Science. And my feeling is to argue against labeling. Just to me, neither pro nor con, just indicates a lack of pride for GMO foods.
Oh, labeling foods.
Yeah.
So, your argument, you're against labeling?
No, I'm actually for labeling because it would show I'm proud of this.
Yeah. No, I think that's a really strong argument for labeling. I think it's the best argument for labeling, actually. That if this isn't something that we think should be demonized, then label it, and it can be a good thing, like pasteurized milk. So, I don't actually have a position on labeling. I think I struggle a little bit with it because why would we label something as if it's dangerous when there's no scientific basis that it's dangerous? But on the other hand, I think that the best thing we can do is label it, and people will get over it, and maybe they'll celebrate it, like we do with pasteurized milk.
Or slightly change your labels. So now you say, instead of saying contains GMO, which is kind of like got worms or something of this sort, you say contains high quality GMO ingredients.
Right.
Emphasizing that this is an improvement.
That save the environment.
Yeah.
Reducing pesticide application.
And if you look at like Arctic apple, for example, I mean and I think DNA potato also, like they're really promoting and branding their product as something special, and it's GM and it's special, and they're branding it as such.
I support labeling for a different reason, because I think it's going to create a tremendous wave of inquiry and questioning, because every state government, every municipality, everybody around the country with huge numbers of lawsuits. The USDA has officially said you can't call things GMO-free because we don't recognize the concept of GMO, right? I mean, we have substantial equivalence. So I think if you allow people to start labeling things GMO-free, it's like the question of natural, unnatural. People are labeling things natural. What does that mean? So I think actually labeling is fine, because the negative fallout if you refuse to label things is, what are you hiding?
That's right. That's my feeling.
Absolutely. So I don't see the downside to labeling. I see no downside to labeling.
But within that, it's a matter of whether it's mandatory or optional labeling. Because if you're making a mandatory labeling of a process, that's the only process we're requiring people to label. Why aren't we saying, this was from radiation breeding? So it's singling out, it'll be the only process that is forced to be labeled, as opposed to Sarah's example is optional labeling.
Well, we still have
Arctic Apple can be proud of it and then put that on their label that it's GE, but do we want the federal government to say that this is the only process we're going to label on the food sample?
Actually, we do label other things by how they're made, like the
Making handouts.
sweatshop-free textiles, fair trade.
Is that federally mandated though?
It's voluntary.
No, it's a marketing thing.
It is very
It's all
voluntary.
marketing, exactly. But the whole labeling thing is not whether we federally mandate.
Oh, mandate.
Versus optional. So pride comes in the optional route where Arctic Optics can say, hey, you should buy this.
John,
because
did you
it's great.
ever manage to succeed in decaffeinated coffee? That's what John
Well,
Stiles tried
this
to
is
do.
one of the problems about doing it outside of a big company, which are always demonized, is that it's a big project. We started working with coffee, and you have to have a transformation system, and if you develop your own, you have to spend millions of dollars getting it all through regulatory apparatus, so you want to buy it from someone else. Well, coffee doesn't work on a lot of them. That's the first problem. And then just everything is raising enough money. It's a different marketing strategy. So you go to Monsanto and say, hey, you want to partner with us? And they go, well, how are we going to make money on this? Because the trees grow forever, And so there's all these kind of things. It's really hard for small companies to do this. But one of the comments I wanted to make is and it came from this study that Florida State put out a while back from, I forgot what the department was. It was like the ag economics guys. Looking to do a survey to figure out, well, how should we label things? One of the things, it turned out that people don't understand anything. One of the things they asked about was something like 33% of the people that they surveyed thought that vegetables did not contain DNA. And one of the other questions, 80% of the people answered that every plant product that has DNA in it should be labeled. 80%. Now, I mean, I don't know. I don't even know where to go with that. Where were these people in science class? But so I don't know what labeling does. And just one other quick comment. I started working with genetically modified organisms really early on, like in the mid-70s, and we saw the same thing in everything back then. I can remember this guy named Jeremy Rifkin. I don't know whatever happened to him, but he was against everything. And I think when products came out in the pharmaceutical industry after, and this went into the 90s, and everybody's aged parent was taking something that, that's pretty good, So I think that risk thing is maybe goes to that. And other things like they came out with like some of the proteins factors for like clotting that were completely GMO made. And a whole generation of hemophiliacs were wiped out by AIDS because they were taking stuff from plasma, and it wasn't. And so I think maybe you need something like that to turn it around. And we thought maybe our coffee would if we could. Then 2008 came up and you couldn't raise a dime anywhere, so that was like. But it's a really difficult thing for. It's amazing that the papaya thing got through, that they could do that on such a shoestring. It's amazing.
That was before Jeremy Rifkin.
No, it was after, way after. He was the 70s.
As a concept, I mean, it was before the controversy when the papaya got through before GMO was a household name and before Greenpeace.
Yeah, it was pretty early on, but there was still a lot of controversy because I spent 20 years at University of Hawaii where the breeder that worked on the project, Richard Manshart, worked, and it's still there. There are still within the last.
It's worse than ever.
Within the last 6 months or so, I was just talking to someone whose father grows papayas or brother grows papayas on the Big Island that were the transgenic ones. Someone came in and chopped all his plants down one long weekend.
Yeah, no, the
Still
situation
there.
in Hawaii is
Still
not a
there.
lot worse
Yeah.
in terms of.
It's a hotbed of growing GMOs because Syngenta, Monsanto, Pioneer, everybody has laboratories there and winter seed production there, and it's also a hotbed of anti-GMO sentiment. So I don't know.
I guess we're still left with a lot of questions. Not what to do. Any other thoughts or comments?
Should
we could spend a long time talking about this topic. Maybe we can talk about it over some refreshments. Ron?
I am now officially endowed with the power to propose that we all go get something to drink, and then we'll do a toast to Peter Davies, who was the man of the hour.
This has been a production of Cornell University, on the web at cornell.edu.